for the kind introduction and inviting me. It's really a pleasure being here, and I'm really stunned by the fact there's so many people on the other side listening to these lectures. And so, yeah, I'll be talking about our ways of simplifying solving problems related to Cryo-EM and model building. So I was working quite a lot on the model building problems, first in MX, then EM at EMB at Hamburg. Now I switched to a small spin-off in Spain. And this, I didn't do that because it's in Spain and it's fun to be in Spain because I work on remote, but I did it for a very interesting technology that they have and the way that they can characterize PPIs on a really large scale. And you will see to my presentation how my idea of using this kind of technology evolved over time. So by presenting different programs I developed, different problems I was approaching and solving, at some point I ended up in the very fundamental problem. So how you should approach the biological problems with Cryo-EM. So I'm not sure if you have seen this. This is a slide prepared for interactive lecture, and I can see the audience. So I cannot really interact with you. And anyway, I'm not sure if you have seen this publication before. It sounds funny. Can biologists fix a radio? But it's not any kind of a joke. It's a very serious publication about the methodology in structural biology and biology in general. And Yuri, over 20 years ago, complains that the way the biologists present their problems, shown on the panel A, it's not really sustainable, doesn't help to interact and solve the problems, and they should approach them more in a way of electronic engineering, like the schema of the radio below. And if you think about what happened during the past 20 years, we really got very close to this picture. So if you think about this kind of interaction networks, every single electronic element over there is a protein, okay? And we know structures of those proteins, and we know how it works, but do we? If you look again at this metaphor, this example, then and the corresponding really devices, electronic elements that should fit into this diagram, then you realize that something is not that easy as it seems. So I'm not sure how many of you could, for example, without Googling, connect a diode into this diagram or even a transistor. It's even more complex. So even though that we know structures of separate proteins, we still cannot connect them easily in this kind of interaction networks. And that's basically the major limitation of the AI in structural biology, because AI solved a very serious problem. So it provides structures of almost all known proteins in separation. Because if you ask the questions about the very fine details of those structures, or large macromolecular complexes, or interaction networks, then AlphaFold is not enough. Even AlphaFold3 or similar programs, they simply don't solve all the problems related to this. So today I'll be talking mostly about the approaches to Cryo-EM, non-standard ones that can help you to address this kind of problem. So solving interaction networks and building models of large macromolecules. I'll be not talking about the fine details because it's mostly related to the protein crystallography, the crystallography in general. But all the programs I'll be presenting also apply to the crystallography and can be used exactly for this purpose. So getting more from the maps that you get using AI than you get from the maps alone. Okay, so I will not giving introduction to the programs I will start talking about, but because I was talking about them for quite a while already, and so I spent a couple of years working on a very common apparently problem in structural biology and crystallography and Cryo-EM, which is identification of, first of all, the sequences of the proteins that you have in the crystal on the Cryo-EM map, of nucleic acid proteins, and then validation of those models fitted into the maps. And validation in a sense of identifying the issues with the sequence assignment. So that's how it relates to the issue with the connectivity, for example, that if you get your sequence wrong in your model, then all the connectivity within your macromolecule will be wrong, and the functional analysis will fail. Okay, so I will start with unknown proteins and go very briefly, quickly through this. So this is not a problem unique to Cryo-EM. So of course, in Cryo-EM, it happens very often. Like this example shown here, these are reconstructions from native cell extracts. I'll show one of the examples later on, or the ribosomal reconstructions when you can find unexpected components or proteins are not fully characterized. But this was quite common problem in the early days of crystallography as well. So you may know that the very first structure solved by Kenji was also sequenced from the map. So it was identified from the density, even though it didn't have technology like we do at the moment. So my approach to this problem was relatively simple and followed the standard procedure, but I used neural networks to make it a bit easier and more robust. So the idea is to analyze the fragments of the map around already built model of the backbone, and then to use very simple neural networks. It's very important, a simple neural network. This moment why it's that to generate something a full footprint of the probabilities of finding different residue types along the backbone that's already fitted into the reconstruction. And this are published. The programs that implement this are available in CPEM, CCP4. Most of them have interfaces. There's not much to talk about that, but I would like to show a few examples how it really works. And that's my favorite one. So I can even show you that I like it so much that I even printed this map because this is a map of the hydrogenase from Chojnowski thermophilum, built by my friend Panos Kastritis from Halle, who is an expert in building cryo-EM reconstructions from native cell extract. And this was supposed to be an easy task, so he built several reconstructions. You can see a few of them here. And then with different approaches with the Omokage search to identify the corresponding proteins by the quaternary structure, by their shape. And then my task was relatively simple. So he asked me to help him to build those models with AlphaFold because this publication was finalized just after AlphaFold was released. So it would be one of the very first applications of AlphaFold for model building at this resolution. But the problem turned out much more difficult than we expected. So it turns out that there are two dehydrogenases, chromium, that have exactly the same quaternary structure, at least on the level of the map resolution that we had, but they're completely different. They share only 20% of the sequence. And so you can imagine that they're really different proteins. And in normal circumstances, it's almost impossible to say one from the other. But it turns out that Find My Sequence, one of the programs I just introduced, can solve this problem easily, assuming that we can provide a model. And at that time, as I said, AlphaFold was available, so I predicted both models with AlphaFold, fitted them to the map, and based on those models, Find My Sequence identified both as only one of the dehydrogenases. So even if the model was slightly off, Find My Sequence still pointed to the correct sequence variant, which we then validated experimentally. So if you can provide a starting model, which is easy nowadays, you can say much more than apparently map provides information. And I also use this program. This is pure AlphaFold release for model building, and it's one of the use cases that you can also find interesting at some point. So this is a problem that we could solve with AlphaFold in no time, but back then it was a very difficult task. So we had this reconstruction of ESX4 hexameric pore complex of type 7 execution system with massive transmembrane region at pretty poor resolution. We couldn't get anything better for it. And the question was how to reconstruct the connectivity of the helices. In order to connect them, I had to identify their directionality and then fill the gaps, the missing loops. And it turned out to be very easy with combining CUT with Find My Sequence. So what I was doing, I was simply fitting the helices with CUT, and then feeding both into the alternative directions. You see that from the map, it's impossible to say one from the other, which one is correct. And then using both as an input of Find My Sequence, and then from the p-value gradient, it was easy to identify the correct one. After this paper was published and after it was released, we could validate that quite nicely, and it it turned out to be completely correct. And I will say in a moment why this validation of AlphaFold is so important. Okay, so if it works out nicely for difficult fragments of the model, then I just thought, okay, then maybe the same approach could be used also for model validation. So we could take fragments of the existing model, ignore its sequence, and try to reassign those fragments to the target sequence and see whether the new sequence, newly assigned one, agrees with the actual sequence of the model. And then there are a few possible cases. So first of all, the p-value or the probability that this newly assigned sequence is completely random can be high, close to 1. Then we know nothing. Then maybe model is wrong, maybe map has not enough information, tough to say. But if p-value is small, then interesting things can happen. So first of all, the sequence, newly assigned sequence can be the same as the one in the fragmented model, which is great. We just confirmed our hypothesis, but the sequence can also differ. And which basically means that we just provided a new sequence alternative to the model that it's more plausible than the one that is an actual model, which means that there's an error. And those errors are amazingly common in deposited models. So and this is a basic idea of program called check my sequence. And but the most important idea behind this program that can validate both nucleic acid and proteins, I will talk a bit more about this in a moment, is that it's designed in a way that's extremely specific. So it will not identify all the errors in your model, all the potential register sheets, for example, very short ones. But whenever it says that there is an error, you can be sure that it's almost that it's there, and everything should be checked. Which means that for very large models, it will produce a very easy to interpret result. So that's one of the examples, early ones. It's a map built quite a few years ago. I think it's quite a few years before the AlphaFold release. And this example shows something fun. So this is the deposited model, fragment of it, that it's register shifted. And I'll check my sequence is is very clear about that. So this bar plot shows the probability that of the different fragment sequence assignment. And whenever the bars are red, it means that this fragment can be assigned with more likely sequence variant different than the sequence in actual model. And I'm not sure if you would dare say whether the sequence is correct or not, because there's simply not enough information in the model. That's probably why our authors of the deposition was struggling to build it. And this one is a corrected model with AlphaFold, where in this particular region of the prediction has very, very high confidence. And again, it's not easy to say which one is correct. This one is correct. And I would like to show you something very important for this kind of analysis, which is comparison with the Q-score. And which in this very case, it doesn't provide enough information to say that there's any error, unlike check my sequence. But it doesn't mean that there's something wrong with Q-score. It simply means that the two programs are targeting slightly different issues in the model. So the way the check my sequence is designed allows for combining probabilities of being misassigned of consecutive residues to a single number, which means that the longer the misassigned region is, the stronger the signal will be, and then more reliable will be the actual answer. Q-score is more designed for local issues. That's why combining the two would provide you the full image of your model and will allow to identify all potential errors, never one alone, I think. And just to convince you that it makes sense to use check my sequence, I will show this example. I keep showing this example for a couple of years already. And it keeps being not validated in PDB. It's not validated because it's too huge. It's an in situ reconstruction of light harvesting mega complex, 40 megadalton complex with 2,000 chains. And it's simply too big to be validated with any of the available programs. Nor MRQ nor nullprobability can provide the scores. So unless I'm wrong, it's still not validated. I checked that last week and it still wasn't. But with check my sequence, owing its extremely simple neural network hard-coded in the program, this structure can be validated in something like 1.5 hours on single CPU. And after this 1.5 hours, it lists below 150 errors. Of course, it doesn't mean that there's only 150 errors, but this is a number that the program, it's 100% sure the errors are there. And believe me, I check them and the issues are real. So the most common issue are so-called unidentified chains, which means that the program could not find for a protein chain in the model any corresponding sequence in the provided sequence set, which means that either the sequence is not there or there's something wrong with the model. In this particular case, the issue was most probably related to the magnification error, and this model is slightly off the density, and simple refinement with self-restraints in CUT brings it back to density, and then check my sequence can identify the model. So it's easy to fix, not easy to find in, remember, 40 metadalton structure. But there are other very interesting models. I will just show you 3 of them. So that's my favorite one again, apart from the dehydrogenase. So it turns out that there are 2 very similar proteins in this megacomplex in the same context that have a quite different sequence, but otherwise they look identical. And the differences between them cannot be predicted and they cannot be identified from the map alone. And they are swapped in the deposited model. And it basically means that the whole environment of those proteins, unless the swap is functionally important, is completely wrong. So the connectivity of those involving those proteins, it's wrongly modeled. And remember, it's a 40 megadalton model. It's extremely easy to make this kind of error, but it's very difficult to spot it and fix. That's another one showing something very standard. So in a low resolution region, the model is register shifted because there is insertion, so one extra residue that affects the identities of all the residues downstream in the model. That's how the deposit model looks like. That's how the alpha model looks like. And now I will start slowly changing gear to the AlphaFold and the use of predicted models in model building. And so from this, you should be able to see that even having AlphaFold model for building this model, it will be not easy to build it correctly because the region which is poorly resolved in the density, it's also not very well predicted by AlphaFold. And this is a very common issue. It's not only about disorder. Sometimes it's simply about the lack of information AlphaFold could use to build the model. And there's lots of this kind of issues in this structure and in PDB in general, unfortunately. And they are not easy to avoid. And that's my third favorite example from this Institute photosystem megacomplex. And it's a very simple problem. Some transmembrane helix, that's how it looks like in the model. That's how the prediction looks like, perfect prediction with pretty high scores. PTM is not very high because it's not a globular protein, nothing unexpected, but PLDT is almost 100, which is great. But when you look at the model or the alpha prediction fitted into the map, you would see something like this. And the phenylalanine sticking out of the map, it's not good. So aromatic residues in Cryo-EM should be in the density, right? Because they are interacting with the beam very strongly, and they should be possible to use them as the sequence indicators, and one should be able to identify the correct register based on them. So something is wrong in here. And what's wrong is that it's not easy to catch, but it's very strongly, nicely visible on those plots here. So this is the sequence coverage from AlphaFold that was obtained to generate this model. And and you can see that there's this gap inside. So this inside, in the middle of helical regions, gap usually means that there's some unusual residue in there. An unusual residue in alpha helical regions would be a proline. And it turns out that this proline simply changes the local geometry of the helix. It becomes something called pi helix with slightly wider radius, and the slightly wider radius changes the register of the downstream residues in the model. But this is not prominently enough modeled by AlphaFold. And that's why it's very difficult to correctly build the structure using alpha prediction. The pi helical region is there, but it's simply not wide enough for real space refinement programs to result in a correctly refined model. And that's something that will be very difficult to address. And this model wasn't trivial to rebuild, even though I knew what's going on. Okay, and so now I will switch to nucleic acids. So with proteins, the easy thing is that in most of the cases, you have at least AlphaFold predictions of separate chains. It's not the case for nucleic acids. So AlphaFold 3 can predict, and Volts and Chai1 can predict some of the nucleic acids, but they're usually not very good models. And forget about ribosomes, they're too big and too complex. And I'll show you this example. That's from a deposited model to make you realize how difficult it is to come up with a correct sequence assignment of an RNA or DNA model. It's simply that, first of all, in Cryo-EM, unless you have super high resolution reconstruction, the sequence effectively reduces to the two types of bases, purines and pyrimidines. And sometimes register shifts, and not sometimes, usually result in very, very few clearly visible outliers. So this region of the ribosomal RNA is massively shifted. So the opposite chains are shifted one residue in opposite directions. And I will show you in a moment how weakly it affects the overall connectivity in the model, even though it's completely wrong. But now the problem is that because the information content of the maps for nucleic acid is so low, and every single one of you who ever built RNA or DNA structure knows it very well. It's very difficult to assign the sequence to the model based only on the map features. There's more information needed. And the idea I had to address this problem was to complement the information with secondary structure information. But it seems absurd, right? So we have a model fitted to density. We don't know the sequence, and you want to get the secondary structure, which is, of course, sequence dependent because it's a pairing of the side chains of the bases in the model. But it turns out it can be done in a very simple way based on the backbone conformations. So I implemented a very simple algorithm that takes fragments of high resolution RNA or DNA structures and fits them onto the unknown backbone-only model with the sequences ignored, and from that identifies the actual connectivity or secondary structure. And just to show you how well it works, I will show you this. So this is a model from RPORP. RPORP is a model building software, very popular in crystallography. My task long time ago was to adapt it to the, not that long ago, but a while ago, was adapted to Cryo-EM. And the problem with RPORP was that it's a hard-coded real space refinement procedure, but no secondary structure restraints. So at lower resolutions, it often happened that the model after model build looked like this. So there's simply not enough information to refine the model without proper restraining. It looks very bad. But it turns out that in this mess, the backbone is correctly placed. And based on the backbone conformation, double helix, the program that I wrote to implement this idea, can identify the double helical region at the base pair restraints, and after short refinement in CUT, you will get something like this. So of course, bases are not resolved in this model, but the backbone almost did not change. So there's lots of information in backbone conformations that you can use. And just to show you a bit more realistic example, that's the famous RNA polymerase. That was a very nice example of cooperation with developers and experimentalists. There were a few errors in the model that were corrected in the subsequent depositions. But one of the issues was the not fully correctly refined terminal base of the nucleic acid fragment. So you see that this is not fully in the density and the connectivity is not optimal. So with double helix, you can get the proper restraints based on the backbone only, and this can be refined to get more plausible model. And this, by the way, corrected and updated the position. Okay, so what about the identification of RNAs? So I will not be showing you examples of this, but they are examples of structures in PDB. The only ones I identified are in crystallographic structures, where complete RNA chains are misassigned, so the completely wrong sequence. And this is, of course, very difficult to identify, and because the information content is very poor. So my idea was that by combining the secondary structure information derived from the backbone conformation and the information from the most plausible type of the base, purine or pyrimidine, can be directly fed into a program called Infernal. It is a sequence alignment program for RNA, and this can be used for the sequence identification. And this cannot be used for the sequence validation. In the procedure I've seen before in Check My Sequence, again with the same procedure, the same p-values, but including also the secondary structure of the small fragments that are reassigned to the sequence. Okay, so I will show you a few examples with a bit more in-depth analysis of the sequence misassignment in deposited models. They are pretty common, unfortunately, but it's, as I said, it's not easy to make them, in particular in large models, and it's very difficult to correct them. So this is a 3.4 angstrom Cryo-EM model of mammalian ribosome, and this one you've seen before. And the reason I selected this fragment of the model to present is that in this model, even after correcting, not that much changes. So this is actually the region where there was this issue with sticking out purines, where pyrimidines should be, but otherwise, the connectivity, the base pairing in the model is almost correct. So I'm not sure if you are familiar with those arc plots that simply show the base pairing in RNA sequence, and this corresponds to the extended double helical region. And so the top sequence, it's as a reference structure, then it's a actual structure that you've seen on the previous slide, and this is a correct. The upper one is corrected one. Sorry. So all the nucleotides marked in orange are the ones where there is a mismatch of the base pairing. There's not that many of them, at least not as many as you would expect from this kind of serious errors. As I said, both complementary chains in the model are shifted by one residue in opposite directions. Most of the connectivity is okay, and majority of the purines sit where the purines should be, and vice versa. So this error is really difficult to spot. And looking at every single residue in the model is not always doable for the experimentalists. And where the error comes from, because also very interesting. So I chased that in this model. And it turns out that the porcine 28S model was built on the earlier structure, low resolution structure of human ribosome, which looked like that. And this one was built combining different ribosomes and using standard modeling programs in RNA world and could. So most probably it wasn't correctly refined, and the base pairing wasn't prominent enough to identify the proper restraints for the refinement, and that's where the shift comes from. So it shows how difficult the task is and how helpful Check My Sequence and double helix can be in addressing this problem. So that's another example of ribosomal RNA with issue. And this one is very easy to spot if you know where to look. So that's the sequence alignment of the model sequence and the reference sequence. And what's peculiar in here is that those two gaps in here. And if you look at the actual model, there are really gaps. So there's some kind of a mismatch in the model that exposes the single base pair that wasn't modeled there. So there should be not extended backbone, but there should be another base in the middle. And that's how the corrected model looks like. And this would also get rid of those peculiar gaps in the sequence alignment. So sometimes, if you know what to look at, you can identify serious error, but looking at very trivial data, you basically get during the position. That's another example of DNA, and that's a nucleosome solved by a very experienced group. So I don't know what happened. Apparently, they must be in a hurry to deposit this. And you see that the model is completely off density. And that's kind of information you would get from check my sequence if you run validation of this program. So the identified chains. And then looking at them, looking how they fit to density should help to address the issue. So in this case, the base pairing is okay, but definitely not correctly refined in the density. There's another error, this one, and it's an obvious register shift. So again, there's something missing in there, and the whole chain is shifted by one residue that ends up in this kind of issue in the feed. Okay, so now I presented you software for identifying register shifts in protein. I mentioned it's a quite common issue, and then RNA counterpart of it. And now you may ask, okay, but now we are using AlphaFold to build the models, fit the components into the map. How plausible it is that those errors prevail? So there's lots of errors in PDB. So maybe also AlphaFold learned those errors and is producing predictions of register shifts. So it turns out to be not the case. And together with Daniel Rigden from University of Liverpool, we performed a very nice study, was guided by him, by Daniel, showing that how good AlphaFold in fact is and how well it avoided this trap of register shifted proteins during training procedure. So it cross-tested our approaches, which is based on the fit of the model to the map. And Daniel's approach that is based on the comparison of the connectivity in the deposited model and connectivity in the AlphaFold predictions. And it turns out that they are perfectly complementary. One works better when there's high resolution information in the map, the other when there is no map available. And by combining the two, we performed a large-scale analysis of the PDB structures. And it turns out that there's lots of register sheets. So sometimes they are small ones in the periphery of the structure, sometimes very prominent ones, sometimes whole proteins are completely register shifted from beginning to the end. And this trend is not really changing much. So it seems that for Cryo-EM, the fraction of register shifted models deposited to PDB is decreasing because the map of the models, resolution is usually decreasing, but it's not really the case for MX structures. Maybe they are simply more difficult and desaturated already at a level that corresponds to the standard error rate. But interestingly, there's not much dependence with the resolution. So it's very likely that simply the availability of the higher resolution maps simply helps to beat the lower resolution maps of similar proteins, and that's why the trend is towards the improvement of the number of errors. Still, from this analysis, it's quite clear that you can trust AlphaFold as would be expected, because AlphaFold is a connectivity-based approach, so it should not be wrong about connectivity, and it's not reproducing register shifts. So if you are lucky and your protein can be predicted with high confidence, you most probably would end up with a very correct model without this kind of issues. Okay, so now the question would be how many ordered proteins you can predict with AlphaFold? And it's a pretty good question that it's very important, not for model building, but also for the analysis of the protein complexes and space of the proteins, structural space of the proteins. So I did kind of a simple study of this based on check my sequence. So I ran check my sequence on all the Cryo-EM models in PDB and identified those for which check my sequence says that certain chain cannot be identified, which means that it sits in a very low density or it's poorly built. And then I checked for how many of those poorly built chains or not in the density, AlphaFold can predict something with high confidence. And it turns out that for most. So just to show you what I mean by the outliers and weak density built models, I will show you this. So this is E. coli ATP synthase, so textbook protein. And it turns out there's only one E. coli protein in PDB for this complex, and one of the transmembrane proteins simply not in the density. So the Q-score, I said the Q-score is a good score, but you need to combine with other ones sometimes, it's very, very low. We basically see this no density for this protein. But also the prediction alpha database of this protein, even though it's very similar to the deposit model, has very low confidence. So we cannot really rely on this. So PTM, PLDTs are all low. So this is one of the examples when our structural there's gap in our structural space. We know nothing about this protein given current status. But apparently, there's not that many of around identified around 2,000 protein chains like this, based on this analysis, which is, I think, pretty convincing that you will not, apart from disordered proteins, of course, face this kind of issue. So switching from the prediction and analysis of the single proteins, I would like to talk about protein complexes now, because that's the most important part. So as I said at the beginning, AlphaFold solved the problem of just determination of single proteins in isolation. It's extremely good in that, but it's not very good in predicting structures of complexes. And just to show you what it means, I copied this picture from a recent paper that was describing the large-scale analysis of the human proteome from structural perspective. And what this plot shows, it's the distribution of P-Q score, which is a meta score for analyzing quality of the predicted models, and varies from 0 to 1. Of course, the higher, the better. And the analysis is based on the plausible complexes in human proteome from HuMAP and HERI databases, existing structures, and models predicted with different kind of evidence and completely random set. For completely random set of protein pairs, of course, most of them are predicted with very low P.Q because they are most probably not proteins, protein complexes. They don't form stable complexes. But amazingly, even for structures, that there's a great evidence that they form stable complexes. Either there exists a structure or two hybrid systems confirm that they interact with each other, cross-link data. There's still lots of structures for which AlphaFold cannot predict those complexes. And now the question is how you can use the information, additional information, speaking today, Cryo-EM maps, complement this missing information, how to get more of the maps when the resolution is very low. But at least you know from the reconstruction that the two proteins form a complex. And to address this, I developed a very simple program. I developed that because I really needed this for quite a few projects. That's called gap trick. It's a very trivial idea, but I think it addresses the problem that is very serious. So the major problem with AlphaFold is that the main source of its success for predicting single chain proteins is that it can use so-called templates. So you can identify similar proteins in PDB and use them to guide the structure prediction process. But this cannot be done for complexes. And there's a very good reason for that, and it's disabled on purpose in AlphaFold. Simply the risk of introducing wrong templates, and it's a very deep topic, it's too high. And it simply wasn't properly tested, and the authors had to disable that. And the input information on interchain contact is simply masked. And there's a program called AlphaFold Unmask that removes this mask from AlphaFold 2 that enables the usage of complexes. But I decided to do something similar, but with AlphaFold trained on the single chain proteins. And I had a very good reason for that, because this approach, first of all, is not biased by the training set, because it has never ever seen a protein complex in its artificial life. And it's also very easy to validate, because I can take any complex out there and use for validation, because none of them was used for training those neural networks. Okay, so what I learned from this simple tool, gap trick. So first of all, I checked how AlphaFold 3 performs for different kind of complexes out there. So I use PISA to identify thousands of stable complexes in PDB. Stable means that the predicted Gibbs free energy delta G should be above 0. And then I removed from those protein complexes, everything except protein components. So the ions, ligands, short nucleic acids, if any, and so on and so forth. And it turned out that that many of those, for many of those complexes, the delta G became negative, which means they're not stable without the presence of those components. And for those weak complexes that require something extra for the formation, AlphaFold 3 very often fails to predict a structure. Very, very often. But the good news is that even though it fails to predict them, it correctly assigns them very low score, which is very good because that's what you expect from AlphaFold. So if it's wrong, at least it should say that it's wrong, and that's perfectly fine. But if you take gap tree, it's very simple stuff, and structurally biased target structure as templates. So I rotated and shifted them randomly. Then these most of those complexes can be reconstructed. So we can build the weak complexes. Are they small? And what does it really mean, weak complex? And so of course, like I said, it can be a complex that is missing some kind of component, ion, small molecule that it's stopping to protein components. But it can also be subcomplex of an overall very stable protein complex. So for example, the tautomerase is a very good example. So I'm pretty much sure that every single one of you predicted structure of tautomerase with AlphaFold because a standard example in CollabFold. But the story about tautomerase is that biologically relevant and stable in solution oligomeric form is a hexamer. Okay, then what's usually predicted as an example structure in CollabFold is a dimeric subcomplex, which is very stable as well. But it's not the only interface in this structure. There's another one, this one below. And this one can be predicted only with gap trick if you use the corresponding template, bias template. I always talk about bias template. So it's something roughly similar to what you expect in a target structure. And but it looks ridiculous, right? So if you have prediction like that, you may start wondering, okay, does it even make sense? Because it's another like a structure in a stable solution. So there's something more. So there are contact predictions based on histogram, so internal data structure in AlphaFold. And I found that they are extremely reliable discriminator of the correctness of the model, and they are extremely reliable overall. So the precision of those contact predictions, regardless of the quality of the actual prediction, is extremely high. So if you have kind of a rough template for Cryo-EM, run it through a gap trick, and you get lots of contacts, you can be sure that the structure is correct. And just to give you some evidence for that, I'll show you very briefly this result of the numerical experiment to confirm this. So I took 300 structures from HUMAP2 database of human PPI. So they provide negative sets, so protein pairs that are very unlikely to form a complex. A positive set, very likely pairs of proteins. And I printed all of them in AlphaFold 3. AlphaFold 3 is hallucinating like hell, which means that if you look at the predictions from the negative and positive set, both complexes in both will look like a proper valid protein complexes. Nicely folded, secondary structure, and so on and so forth. On the score will be relevant. But it turns out that gap trick, based on the contact prediction, can very nicely discriminate between the two. So there's such a significant difference between the number of contacts in one and the other. And if you take structures that are known to form a complexes, and this actual structure solved, this is from the medium and low resolution EM models, then the number of contacts explodes. Because for this, there are stable, strong complexes for which the evidence that they form complexes is very, very strong. And this, looking at the ROC curve, it's a very good discriminator of the plausibility of a complex. Okay, maybe I skip this. So this is an analysis because I'm running out of time. Maybe not yet. I will very briefly go through that. So, but then you may wonder if you're running your structure, initial structure, tentative structure from built into Cryo-EM through gap trick, how good will be the resulting model? I mean the geometry, how the sidechains will look like, the packing and interface will look fine. And a good way of validating that is running that through PISA, because PISA is a software originally developed for crystallographic models to identify biologically relevant assemblies in the crystal structure. So it's very sensitive to the quality of the interface. And in this test, I rebuilt with GapTrick high resolution and good quality crystallographic models, which means that I have the Gibbs free energy of the reference models very well estimated. And it turns out that GapTrick from AlphaFold can reconstitute those interfaces that I used to calculate the delta G with very high confidence, but only if you have high confidence predictions. So in cases like this, mostly disordered, poor PTM and PLDGT, most probably the interface will be also bad. So that's a kind of a trivial observation, but I think it's important because this ongoing discussion whether using PISA for in Cryo-EM and AlphaFold makes sense. It makes if you have very high quality predictions and models. So, but I said at the beginning that I developed GapTrick for a very specific purpose. So I wanted to use it to complement the missing information in the low resolution maps. And this is one of the projects. I cannot say you much about it because it's still unreleased, but it's a longer story. And that comes from this map to this one in roughly 2 years. So because I had GapTrick, I could beat initial models, identify the contact, like this cluster of salt bridges, and perform I'm not doing experiments, so the authors of the structure performed the functional analysis before we got the high resolution structure that allows for the direct identification of the contact. So that's how GapTrick can be used in a very difficult Cryo-EM cases. And that's an example from PDB. And this is a very important oncogene. And unfortunately for this one, there's no high resolution structure. But there's a hybrid of human and rat protein for some reason, but not a purely human complex. So this is a phospholipase kinase complex. And the mechanism of this kinase is very poorly understood. So what people believe is that it binds to the phospholipase, and then using some kind of far-reaching hinge movement, phosphorylates tyrosine in the remote location. So that's the Cryo-EM structure, that's the map. So you cannot say much. All you can do with this is to fit the Cryo-EM AlphaFold predictions, but you don't see really fine details. But it turns out that running this model built from AlphaFold predictions through GapTrick reveals the most crucial contact in the structure and very, very weak one. So this is a tyrosine that needs to be phosphorylated in order to be tightly bound by the charge pocket in the phospholipase that then allows for this hinge movement and actual activation of the enzyme. And this would be impossible to get from the Cryo-EM data map alone. And as I said, the crystal structure doesn't exist for this specific variant. But there's something more. So GapTrick can do something super convenient. So you can also rebuild not fully refined or difficult to refine models to get better models. And this one, for example, owing the resolution, is not a perfect one. So of course, there are no Ramanujan outliers. That's obvious because most probably they used Ramanujan outlier trains, but there's lots of clash scoring here. But we're running that through, you can check in the paper, running that through GapTrick produces a perfect model. Of course, you can say it's maybe biasing the model because it's a prediction and so on and so forth. But you should remember that usually the not energetically favorable conformations, so with bad geometry, speaking of other words, in the models, are allowed if you have very good evidence that it's important. Otherwise, they should have perfect geometry, and GapTrick allows to get this. So the models you run through GapTrick in most of the cases, not always, you have very good Ramas that score very few Ramanujan outliers, or none at all, and no Rotamer outliers and very low clash score, which means that it will be very easy. And that's what I have been doing for a couple of last years at EMBL. run those predictions, for example, through server code, without any restraints like higher order restraints, like Ramachandran plot restraints, and you will get perfect models because the models will be in the map will be already pretty well relaxed. There will be no strains in them, and that works very well. Okay, so now yesterday someone asked about disordered proteins, and again I would like to tell you a very short story about disordered proteins and the contact analysis. Because I can show it because it was the story was released 2 days ago, and finally I can talk about that. So this is about proximal protein interactions, and it's a difficult cryo-EM study, as you will see in a moment. So peroxisomes, they're organelles without their own genetic code, so they need to import all the machinery from outside, and they use so-called transporters for that. And this is not really understood how they import with down the pores, how they release the cargo inside, and so on and so forth, how the transporters are recycled. And in one of the studies, we try to understand how one of the transporters, PEX5, interacts with PEX8, which is both cargo and the transport cargo releaser. But it works in a very peculiar way because it doesn't rely on the standard interaction motif between the transporter and the cargo, so-called PTS1 motif, that is at the end of a longer peptide, which is very important because it means that the coordination of the transporter protein relative to the cargo is very poor. That's why it's a very, very bad target for a cryo-EM study, and it's even worse target for crystallographic analysis. And on top of that, it's a relatively small complex. It's of the ordered mass, it's roughly 100 kilodaltons, so it's really small stuff. And so that's the kind of a map we could get at 5-function resolution. We can easily interpret most of the stuff, like the position of the PEX5, PEX8, lots of disorder stuff around, and it's very strange thing here. And it turns out this very strange thing here is a crucial part of this model because it's a secondary interaction motif. So, but in order to analyze this, we have to build a model first. And it's a disordered protein. And we also wanted to show that this disorder makes sense in solution. So now kind of an artifact. So in this study, and it's quite a long story, so I'll cut it short. We combined the Sachs analysis to build an ensemble of conformations of transporter related to the cargo. Then I rebuilt those models with gap tweak and fitted all of them to Cryo-EM to show that all of them fit equally well or equally bad. Because the models shows the high disorder of the transporter protein relative to PEX8. And most importantly, from the alpha analysis and gap trick, we also got a very strong evidence that a small blob in the map, shown here, it's in fact a short helical region in the middle of the disorder, a part of the transporter protein, that it's reaching over the whole PEX8 complex to form a very strong interaction over there. And then, of course, the biochemical analysis followed to prove it's true, but this evidence comes initially from the contact prediction based on AlphaFold, not from the experiment, because we did not have enough evidence from the map, low resolution, or other biochemical data like crosslinks to show that it makes sense and to explain this small feature in the map. So I hope I convinced you that using the contact analysis and gap trick really boosts the information content that you can get separately from the predictions and Cryo-EM maps. Okay, and with that, I would like to thank you for listening and mention people involved in the project. There's quite a few of them. So all the projects I was presenting are the close cooperation CCP4 and CCP-EM teams. Dan Riden group was working with them on the validation projects and register shift analysis. Panos, as I said, is an expert in native cell extracts, so we have very funny analysis of the unknown proteins in Chetomium. And Matthias Willmann's group and lots of people from EMBL Hamburg and EMBL Heidelberg were involved in working on that. And all the programs I shown you are they published, like Find My Sequence, Check My Sequence, Double Helix. GapTrick is released. It should be accepted soon, I hope, in bioinformatics, but it's fully available. And because it relies on AlphaFold 2, which is not trivial to install, you can run it in Google Colab without running anything, installing anything on your computer. So it's very convenient, and I really suggest you to use it for refining your models in difficult cryo-EM cases. Thank you.
All right, thank you, Greg, for a great talk. Got time for a couple of questions. For Find My Sequence, does one need coordinates in PDB or MMSIF format, or can one start with just a map and a sequence? And maybe more generally, when would you advocate for people using these tools? Earlier in the model building and refinement process, or more as a check towards the end?
Yeah, it's a very good question. So Find My Sequence relies on the actual model, like all the tools I've been showing. But they don't need to be very accurate, which means that you have any guess of the backbone model that possibly fits the density. You can get it from fitting AlphaFold guess of the structure, or you can also combine it with tools like Model Angelo. I'm not sure if you have a talk about that. So it's a really good model building software, modern model building software that can do great stuff. But it seems from the publications that people in difficult cases get best results combining this with seemingly much simpler Find My Sequence. So they build initial model with Model Angelo, then refine it manually, and then run Find My Sequence in difficult places, just to identify missing fragments or get the connectivity. But what I would suggest you to run really early on during model building is Check My Sequence, because it's very fast. It's hard-coded in Doppio in case you use it. And it can very nicely guide you to the model building and refinement process. So you'll notice, for example, that the p-values will get lower during the model refinement. The models parts that are identified at the beginning will be identified at the end. So it will really help you to focus on difficult regions, and it produces very simple reports. So you just need to read the text answer. Check this region of your model is probably wrong, and you simply go there and fix. And I like it very much, and it helps avoid issues during deposition.
All right, a couple of related questions. Are these tools available as part of the CCP4 or Phoenix packages, or are they completely separate? And if they are, how does one find or run these tools? Are there web servers or binaries available?
Yeah, so Find My Sequence, Check My Sequence, and Double Helix are cross-platform standalone programs distributed with CCP4 and CCP-EM. So not all of them have graphical user interfaces. So Check My Sequence does, for example, in Google Cloud. Find My Sequence as well. Double Helix doesn't, but Double Helix works mostly in the background of Check My Sequence. So if you can use, that's why you can in CCP4 and CCP-EM, you can easily validate structures of protein nucleic acid complexes. Just need to provide the correct sequence variants. So, and yeah, so this will be only CCP4 and CCP-EM, and apart from Double Helix, most of them have GUIs, simple ones.
All right, we've got one kind of hypothetical question here. Say one had a 2.8 angstrom cryo-EM map of an enzyme, and that enzyme contains a loop that's never been modeled before. What would you suggest being the best software to use, AlphaFold or whatever, to fit or to create a model for that loop?
Yeah, so my suggestion, and it's really based in my experience, is run GapTrick on that and see what happens. And very often you will have dominating conformation of this loop from AlphaFold and from GapTrick through AlphaFold. The idea is that by having anchors and fully refined rest of the model, you will guide the neural networks in AlphaFold to build something sensible in this region. And so you can use GapTrick to complete models from automated model building, like Model Angelo. You can run it to build missing loops. or you can use it to build whole domains. And it's not equivalent to downloading the alpha predictions and fitting them to the map, because they can have completely different conformation, not compatible with the map. And after refining the model and using this model as an input to gap trick, you will ensure that the output will have the same conformation, very easy to fit to the map, unless there's something in your model that contradicts the information in the evolutionary information MSAs. It happens. And then it's more deeper study.
Okay, well, end on one last question. Are there tutorials available in addition to the software?
Yeah, so check my GitHub and GitLab webpage. Fortunately, there are two of them. And there's always in-depth documentation. You can always drop me a line and ask questions. So I'm always happy to ask them complaints if it doesn't work. So the documentation should be good enough, but it's not always good enough for everyone. So just ask, complain, and check my GitHub and GitLab web pages.
All right. Thank you again, Greg.
Thank you. Thank you all.